Overview. In this chapter, we shall look at general principles of human genetic variation and how variation in DNA relates to the variation of the sequences found in proteins and non-coding RNAs. Learning objectives for this module are as follows. Following completion of this module, students will be able to explain the origins of DNA sequence variation. Students will be able to summarize the principles of DNA repair. Students will be able to describe the scale of human genetic variation. Students will be able to interpret the functional consequences of genetic variation and protein polymorphism. Students will summarize the genetic diversity of the human immune system. Module 1. Origins of DNA sequence variation. The learning objective for this module is for students to be able to explain the origins of DNA sequence variation. That is, how do variations in DNA sequence arise? Let us begin with some basic principles that are critical to your understanding of human genetic variation and will enable you to read about genetic variation in the scientific literature. The first term is locus. This is a very fundamental term, and it refers to a unique chromosomal location which defines the position of an individual gene or DNA sequence. So it really helps us to localize where a particular genetic sequence is, whether that is a sequence of nucleotides or whether it's referring to a gene. It's that particular location within a given chromosome. Then within a locus, one can speak of alleles. And probably this is a term that you have heard about in the past from your general biology courses. An allele is an individual version of a gene or a DNA sequence at a particular locus on a chromosome, and it's often used loosely to describe the encoded protein variants. So when we talk about the given genotype of an individual with respect to their alleles, they can either be homozygous or heterozygous. Each allele is inherited in a Mendelian fashion from the parent, and so if the two alleles at a given locus are identical with respect to their DNA sequence, then the individual is said to be homozygous. If they are different variants of the same allele, that is, that they differ in sequence, then the individual is said to be heterozygous. And this is a very important concept to understand. Homozygosity and heterozygosity are very important terms. The next concept to consider is genetic variation that does not specifically refer to variation that is inherited. So in addition to the genetic variation that we inherit, that is present in all of our cells, which is constitutional variation, so a variation that is inherited and is present in all of our cells is referred to as a constitutional variation. But in addition to that, there are DNA changes that can occur in the DNA of our cells throughout our lives, or these are referred to as post-zygotic or somatic changes. So for example, if you spend too much time out on the beach and expose yourself to ultraviolet radiation, then that can lead to changes in the DNA of your skin cells, and that would be an example of a somatic mutation. Whatever changes took place in those cells would not be present constitutionally, that is not throughout your body, and rather they would be referred to as somatic genetic variations. And then, there's the notion finally of genotype versus phenotype. So individuals differ from each other at a DNA level, and that is referred to one's genotype. But the observed characteristics that we see in an individual that are the result of genotypic variation is referred to as the phenotype. So, for example, if we were talking about a clinical situation and there is a particular variation at the DNA level that causes the disease, then we would say that this particular genotype leads to a particular clinical phenotype. So, sickle cell anemia genotype leading to sickle cell disease. Origins of variation. Probably a term that we should describe firstly when we talk about origins of variation is the word mutation. One should keep in mind that the term mutation can refer to a localized change in the base sequence of a DNA molecule, and that's probably a very common way of thinking of the word mutation, but it also sometimes is used to describe the process that creates it. So when we think about origins of DNA variation, they can arise as the result of endogenous errors in chromosome and DNA function. So that means that something that's actually the normal nucleic acid transactions that take place in our cell, such as DNA replication. There could be errors in DNA replication that lead to genetic variation or mutations. And there can be errors or alterations in chromosome segregation and recombination errors that again give rise to what we think of as a mutation. Secondly, we can have genetic variation or mutation as a result of exposure to either endogenous or exogenous sources. And that typically refers to some type of chemical. I think this is the most common way that people think about the origin of variation. But it is important to keep in mind that there are basic cellular mechanisms that deal with DNA replication or chromosome segregation and recombination that can give rise to genetic variation. So going back to endogenous and exogenous sources, these types of origins of genetic variation refer to the exposure to chemicals, typically, or chemical environments that then in turn will give rise to alterations of the sequence of the DNA or alterations in the DNA molecules themselves. DNA replication errors. General errors in DNA replication are fairly unavoidable. Each time the DNA of a human diploid cell replicates, 6 billion nucleotides need to be inserted in the correct order to make a new DNA molecule. Not surprisingly, DNA polymerases on occasion insert the wrong nucleotide, which results in mispaired bases, a base mismatch. And the likelihood of such an error simply reflects the relative binding energies of correctly base pairs and misbase paired. In the majority of cases, however, there is a repair process that is in place to take care of mismatched DNA. So mismatch by the DNA polymerase is taken care of through the mismatched DNA repair. The majority of DNA polymerases engage in replicating our DNA have an intrinsic 3' to 5' exonuclease activity with a proofreading function. If, by error, the wrong base is inserted, the 3' to 5' exonuclease is activated and degrades the newly synthesized DNA strand from the 3' end. Then, the DNA polymerase resumes synthesis again following insertion of the correct base. Another type of replication error commonly occurs within regions of DNA where there are short tandem repeats. If, for example, the DNA polymerase encounters a 30 nucleotide sequence with 15 sequential repeats of AT dinucleotide or 10 sequential repeats of the CAA trinucleotide, there will be an increased chance that during DNA replication, a mistake is made in aligning the growing DNA strand with its template strand. A frequent result is that the template strand and the newly synthesized strand pair up out of register by one or sometimes more repeat units, causing what is known as replication slippage. Errors like this are often repaired successfully by the DNA mismatch repair system. Chromosome segregation and recombination errors. Errors in chromosome segregation result in abnormal gametes, embryos, and somatic cells that will have fewer or greater numbers of chromosomes than normal, and thus would have altered numbers of whole DNA molecules. Changes in chromosomal DNA copy number are not uncommon. If they occur in the germline cells, however, they can cause embryonic lethality or lead to a congenital disorder, such as Down syndrome. Down syndrome is commonly caused by an extra copy of chromosome 21. However, changes in copy numbers of sex chromosomes are more readily tolerated. Changes in chromosomal DNA copy number are a common feature of many cancers. Various natural errors can also give rise to altered copy numbers of specific sequences within a DNA strand that may range up to megabases in length. They can occur by different recombination and recombination-like mechanisms in which non-allelic, but often related, sequences align so that the chromatids are paired with their DNA sequences locally out of register. Subsequent crossover produces chromatids with fewer or more copies of the specific sequences. Damage. DNA, as we know, is a comparatively stable molecule. Nonetheless, there are constant threats to its integrity that lead to the breakage of covalent bonds within the DNA molecule or inappropriate reaction leading to bonding of different chemicals to the DNA molecules themselves. Most of the DNA damage, surprisingly, arises spontaneously within the cells due to the normal metabolism generating reactive species that can actually interact with the DNA. A minority of DNA damage is induced by external sources. Chemical damage to DNA can involve the cleavage of covalent bonds in the sugar phosphate backbone itself, leading to single strand or double strand breaks. Double strand breaks in terms of harmfulness to the cell are more significant, as single stranded breaks are more easily repaired. Another way that DNA can be damaged is by the removal of a base. Bases are deleted by cleavage of the N-glycosidic bond connecting a base to a sugar. Alternatively, the bases themselves may be chemically modified in some way. Many base modifications involve replacing certain groups on the bases or adding a chemical group, such as a methyl or larger alkyl group. And sometimes other larger chemicals, bulky groups, can be added on to the base themselves. Sometimes base modification involves the formation of covalent bonds between bases, and that's known as cross-linking. That can happen due to exposure to radiation, such as UV radiation from sunlight. Chemically modified bases may block DNA or RNA polymerases and cause base mispairing if not repaired, and thus they induce mutations. This slide depicts DNA strand breakage. A single strand may be broken by simple cleavage of the phosphodiester bond, as shown in I, or by more complex single-stranded break, as shown in II, in which the ends are damaged and sometimes one or more nucleotides are deleted. On the far right is shown the double-stranded DNA breaks, and these occur when both strands are broken at sites that are in very close proximity. Shown in this slide are examples of base modification. Altered bonding or added chemical groups are shown in red. Examples are 8-oxoguanine, which is shown on the far left, which base pairs to adenine and so induces mutations. Further to the right is thymine glycol, which is not a mutagen but blocks the DNA polymerase, and a DNA adduct, shown to the far right, which is formed by covalent bonding. In this case, of an aromatic hydrocarbon such as benzyl pyrene to N7 of guanidine residue. DNA damage. Most of the chemical damage to our DNA arises spontaneously and is unavoidable. Every day, under normal conditions, lesions are generated in the DNA of each of our nucleated cells. There are three major types of chemical change which can occur: hydrolytic damage, reactive oxygen species, and aberrant DNA methylation. We will consider each of these in turn. Hydrolytic damage. Hydrolysis is inevitable in the aqueous environment of cells. It can disrupt the bonds that hold the bases to sugars, cleaving the base from the sugar to produce an abasic site. Loss of purine bases. Depurination is particularly common. Hydrolysis can also strip amino groups from some bases, leading to deamination, and as a result, the base is left with a carbonyl group. Cytosines are often deaminated to give uracil, which then base pairs with adenine. Adenine is occasionally deaminated to produce hypoxanthine, which effectively behaves like a guanine by base pairing with cytosine. Oxidative damage. Normal cellular metabolism generates some strongly electrophilic and therefore highly reactive molecules or ions. The most significant are reactive oxygen species, or otherwise known as ROS, formed by the incomplete one-electron reduction of oxygen, including superoxide anions, hydrogen peroxide, and hydroxyl radicals. Reactive oxygen species are generated in different cellular locations and have important roles in certain intracellular and extracellular signaling pathways, but they are mostly originated in the mitochondria, where electrons can prematurely reduce oxygen. Endogenous ROS attack covalent bonds in sugars, causing the DNA strands to break. Some base derivatives are highly mutagenic, such as 7,8-dihydro-8-oxoguanine, which base pairs with adenine. The reactive oxygen species can attack not only the DNA strands, but the bases as well. Aberrant DNA methylation. DNA sites that are susceptible to spontaneous hydrolytic and oxidative damage. Each day, every nucleated human cell loses 5,000 or more purines, A's and G's, and about 300 pyrimidines, C's and T's, as a result of hydrolytic attack. The N-glycosidic bond connecting a base to its deoxyribose sugar is cleaved by water for the reaction. In deamination, the amino group is replaced by an oxygen atom to give a carbonyl group. About 100 to 500 cytosines are replaced by uracil in this way each day. A smaller number of adenines are replaced by hypoxanthines. In addition, normal metabolism generates reactive oxygen species that cleave certain chemical bonds, not just in bases, but also in sugar residues, causing breakage of DNA strands. External mutagens. A minority of the chemical damage to our DNA is caused by external agents that can induce mutation and are known as mutagens. These include radiation, such as ionizing radiation that is found in X-rays and gamma rays. So when you go and have an X-ray, and this is why now physicians limit the number of X-rays that you're exposed to because it can actually be harmful. Gamma rays, which are from nuclear fallout, as well as non-ionizing radiation, such as the ultraviolet radiation found in sunlight. Ionizing radiation, such as X-rays and gamma rays, interact with the cellular molecules to generate reactive oxygen species that break the sugar phosphate backbone and cleave the DNA strands. Non-ionizing radiation, such as ultraviolet radiation, cause covalent bonding between adjacent pyrimidine bases on a DNA strand, such as thymine dimers. There's also exposure to environmental chemicals. Some chemicals interact with cellular molecules to generate reactive species, and other chemicals can covalently bond to the DNA, forming a DNA adduct that may be bulky and cause distortion of the DNA helix. Cigarette smoke and automobile fumes, for example, have large aromatic hydrocarbons that can bond to DNA. Electrophilic alkylating agents can result in base cross-linking. Of particular importance to those of us that are working in the laboratory sciences are being mindful of the chemical exposures that could potentially damage your DNA in the laboratory. Ethidium bromide forms a adduct with DNA and thereby is a mutagen. So one should be very cautious in handling that and avoid exposure.